Welcome to ASCP Medical Microbiology. Well, key characteristics that distinguish bacteria, which are prokaryotes, from eukaryotic cells. Bacteria are unicellular and lack nucleus, nuclear membrane, and cell components. Bacterial genome, nucleoid, single, closed, circular chromosome of double-stranded DNA. The plasmids, they are small, circular molecules of extra chromosomal DNA that often carry antibiotic-resistant genes. Gene exchange can occur via transformation, transduction, or conjugation. Reproduction. The bacteria reproduce asexually by binary fission. Prokaryotes do not reproduce sexually. Energy. Energy generation is associated with the cytoplasmic membrane through the electron transport chain. The typical size of bacteria is 0.2 to 2 microns in diameter, 1 to 6 microns in length. The four basic morphological types are cocci, that is, spherical shaped cells, and the bacilli, rod shaped cells, spirella, the spiral shaped cells, vibrios, are the comma shaped cells. Three major nutritional needs for bacterial growth: the carbon source for the synthesis of cellular constituents, the nitrogen source for protein synthesis, and the ATP is for energy, that is, for the cellular functions. Optimal pH for most bacterial growth is 7 to 7.5. The acidophiles grow at acidic pH, alkaliphiles they grow optimally at alkaline pH. The optimum growth temperature for most human bacterial pathogens are 35 degrees Celsius to 37 degrees Celsius, the temperature of the human body. Six bacterial classifications based on atmospheric requirements. Obligate aerobes that require oxygen for growth. The obligate anaerobes cannot grow in the presence of oxygen. Aerotolerant anaerobes can grow in oxygen but prefer anaerobic conditions. Capnophilic bacteria that require 5% to 10% carbon dioxide for optimal growth. The facultative anaerobes, they grow both in aerobic and anaerobic environments. The microaerophilic bacteria, they grow best in reduced oxygen levels. Fermentation is bacterial metabolism, an anaerobic process where bacteria catabolize carbohydrates to produce energy, carried out by both obligate and facultative anaerobes. The final electron acceptor is an organic compound. Respiration, which is oxidation in bacterial metabolism, an efficient energy generation process used by obligate aerobes and facultative anaerobes. Molecular oxygen is the final electron acceptor, more efficient than fermentation for energy production. Bacterial cell structure. Function of the bacterial cell structure. It serves as an osmotic barrier, can be a site of antibiotic action. An intact membrane is essential for bacterial viability. Gram-positive and gram-negative bacterial cell wall. A gram-positive bacterial cell wall has a thick, rigid peptidoglycan layer, which is a site of action for penicillins and cephalosporins. Contains teichoic acid and lipoteichoic acids, which are unique to gram-positive bacteria. A thinner peptidoglycan layer is unique to gram-negative bacterial cell wall. It contains the LPS layer, which is an outer lipopolysaccharide layer, which acts as an endotoxin and contributes to virulence. Endotoxins can cause shock, sepsis, fever, disseminated intravascular coagulation, which is DIC, and leukopenia. Bacterial capsule. It's made up of polysaccharides, prevents or inhibits phagocytosis. In Streptococcus pneumoniae, the capsule is the main determinant of virulence. Pili are fumbriae. The short hair-like structures on bacterial surfaces help bacteria attach to target cells. Example, Neisseria gonorrhoeae uses villi for attachment. Involved in DNA exchange during bacterial conjugation. Bacterial flagella. Determined motility can be used for bacterial classification. Endospores, a survival mechanism that makes disease control difficult. Two spore-forming genera of medical importance are Bacillus and Clostridium. Visit us back for more videos. Subscribe, like, and share. Comment. Thank you.